Okay, welcome to the webinar. Thanks everyone for attending. Today we are going, Jason Hupp, our presenter, he's a field application scientist. He's going to present a webinar on complementary leaf physiology measurements, the LI-600 and LI-6800. Before he gets started, just a couple of housekeeping items. We have a number of panelists that are going to help with questions throughout the webinar. They are Ian Smiley, an application scientist, CC Hall, applications analyst, BJ Clark, a technical sales associate, and Juan Michelli, international operations manager. And we also have Brent Clausen, director of our international group on. So if you have questions throughout the webinar, we have a number of people who can answer them. It's best if you try not to post them to the chat. It's easier for us to handle those questions if you post them directly to the Q&A section of the webinar. Other than that, Jason Hupp, our presenter, will share his screen and get started. At the end of the webinar, we'll have a little bit of time for questions as well. Anything that didn't get answered from our panelists, we can spend a few minutes at the end of the webinar answering questions as well. So sit back and enjoy the webinar, and welcome.
Hello, everybody. Hopefully folks can see my screen. I see Rod's still unmuted, so he could tell me if that's actually showing up. Or maybe he won't tell me.
It is, your screen is live.
Awesome. Okay, well, I'm going to be your speaker today. I'm an application scientist here at LI-COR. I've done a lot of work with the 600 and the 6800. And between my field assistant that I'll introduce as we go through the presentation, I've done a fair amount of data collection between the two that demonstrates sort of how you might use them together. And for those that might not be familiar, we'll just start with what these instruments actually are and a little bit about what they measure. So we make two combined gas exchange and chlorophyll fluorescence instruments. That's the LI-600 Porometer fluorometer that the young lady in the image is using, and the LI-6800 Portable Photosynthesis that the young man here is using. These instruments they operate in fundamentally similar manners, and they measure some parameters that on their surface seemingly overlap. One doesn't serve as a replacement for the other. They do have their own sort of unique niches that you'd use them for sort of as isolated instruments, but they really make a powerful duo when you put them together. As I said, the 600 porometer fluorometer is a combination of a steady state porometer that gives us a measurement of apparent transpiration from the leaf, and from apparent transpiration we get an estimate of stomatal conductance to water vapor. The porometer part of the instrument is on the lower side of, or the lower half of the instrument, and so it's measuring the bottom of the leaf. And then paired with that on top is an open fluorometer that's probing chlorophyll fluorescence under sort of the ambient light conditions. And from that, we can get things like FE/FM if we're measuring fluorescence in the dark, or 5PS2 if we're measuring it in the light. And the instrument's got an integrated PAR sensor, so from 5PS2, we can derive an electron transport rate. It's a pretty straightforward instrument. It's all self-contained, operated with a single hand. And it's really amazing in one key aspect, and that has to do, I mean, it's amazing in a lot of aspects, but it's really amazing in one key aspect, and that has to do with the speed of the instrument. So the plot that I'm showing here is just some data I collected where I took a 600 and I clamped on an artificial leaf. And it looks more or less the same if you do this on a real leaf. This is just the data set that I had handy for this talk. And time zero on the x-axis is when I actually closed the clamp onto the sample material. And you can see in about 3 seconds, stomatal conductance here is a steady state value. It's not changing. That means the instrument has come into equilibrium and we're measuring what's actually happening from our sample. And that happens so quickly here, in part because of the design of the instrument. It's a small volume cuvette that has proportionately high flow rate for that volume. We use some really high precision sensors that we dramatically oversample with a really nice A to D design in the instrument. And so it winds up being that it has a really short washout time. Really high precision means low noise. We don't need much data to get a nice, stable, clean value. The other thing that makes the instrument really fast in the context of a leaf measurement is that it's an ambient environment instrument. Unless you as a user do something silly to change the environment the leaf is in, like flip the leaf upside down or rip it off the plant or shade it with your body, you're not changing the environment the leaf is seeing when you put a leaf in the chamber or in the cuvette. And so it doesn't have an opportunity for the leaf physiology to respond to the measurement. And so we use the 600 in particular for a particular kind of measurement we call survey measurements, where we want to get a snapshot picture of what the plant material is doing under its current environmental conditions. And I'll show some survey data as we go through here and talk about sort of what we would use that for. But we'll introduce the 6800 first. So the 6800 is a full-blown portable photosynthesis system. You can see it's a lot larger than a 600. It requires multi-handed operation. It combines both a carbon dioxide and a water vapor measurement. So from the leaf, we can derive things like the carbon assimilation rate, either net or gross, dark respiration rate, stomatal conductance to CO2, the leaf intercellular CO2 concentration, CI, and from the water vapor measurement, we still get apparent transpiration and stomatal conductance to water vapor. It's a very different instrument from the 600 in respect to the leaf environment. You can see here there's a bean leaf clamped in the instrument, in the cuvette of the instrument, and we're enclosing the leaf and we're controlling the environment around the leaf. And the way that I show the 6800 here, it's got a chlorophyll fluorometer on the upper part of the leaf cuvette. That gives us similar parameters to the 600, but because it's a controlled environment where we control both light intensity and to some degree spectral composition, we can derive other fluorescence parameters like this FV prime over FM, sort of the maximum quantum efficiency under the current conditions. We can get at things like NPQ or QL that tells us about the state of the reaction centers, and we can do a whole bunch more. This is a full-featured fluorometer. The instrument console in the 6800, I mean, it has the user interface stuff. It's where we see data in real time. It's where we push buttons and record data, and it powers everything. But it also serves for air conditioning. So it has the hardware that controls the carbon dioxide and water vapor concentration in the air that's going through the instrument. So instead of making the measurement under ambient conditions for those particular gases, we're manipulating them, and then we can control them at the leaf surface in the cuvette. In the sensor head, we have all the sensors that make the measurement work, and we have a temperature control module that lets us control the leaf temperature as well. So while we can use this instrument to make survey measurements, we can go measure the conditions the leaf is under, and we can set the environment in the instrument to match that. What the 6800 is particularly good at and really well optimized for are these things that we call response measurements. And this is where we hold all of the environmental parameters that the leaf is seeing constant, except for one parameter, and we iterate over that parameter and look at how the physiology responds. And so I have two example response curves up here, although there are other kinds you could do. These are certainly the most common. And this is the carbon assimilation response to light intensity, what we call Q, or an AQ curve or a light response curve. And then the carbon assimilation response to the intercellular CO2, what we call an ACI curve or a CO2 response curve. And these curves are particularly interesting because they're not, if you do them right, the answers that you get from them are not subject to the current conditions the leaf is seeing, but rather they're allowing you to probe physiological capacities and limitations of what's happening physiologically. So you can derive like maximum assimilation rates. You can get enzymatic efficiencies from the ACI curve. You get information about nutrient status from the ACI curve. You get information about how the plant uses energy from these light response measurements. So they're they're much deeper in information than just the outputs you get directly from the instrument. The trade-off for these kind of measurements is that they are incredibly slow, particularly compared to the kind of data we collect with a 600. Where I might take 5 or 10 seconds to make a single measurement with the 600 in a survey context, a light response curve might take me half an hour to an hour to make a single curve. And maybe the same for a CO2 response curve on the shorter side of that, maybe 30 to 45 minutes. So they're not high throughput measurements. But these two different sort of measurement sets lead us to maybe the first way that we can think about using a 600 with an LI-6800, pairing them together. And it's what I've called a sort of a screening and evaluation program here. And this is just a simple model to illustrate the point. My gray boxes you can think of as a collection of plants that we want to explore. So this could be a population of breeding lines. It could be some selection of commercial varieties. It could be a bunch of different accessions, or anything that represents some variability and a large set of it, right? We're not talking about a situation where you have 1 or 2 of these lines that you want to look at. There might be 10s or 20s or hundreds of them. And obviously, as much as we would like to have really detailed physiological information on 10 to 100 lines, right? That's really impractical to do with the 6800, especially a single 6800, because it takes a long time to do those sort of response measurements. But the 600 is quick. We can do really broad scale screenings with this thing that allow us not only to get measurements on all of the lines, but we can generally do them with enough density that we can actually capture sort of temporal dynamics to those behaviors. And in this screening evaluation program concept, we would use that broad patterns observed sort of under ambient conditions with the 600 to identify sort of outlier lines. Those might be the lines that respond the most to some particular stressor we apply to them or some particular treatment, or the ones that respond the least, or maybe the combination of the two. And we can use that sort of temporal data that we've gathered to guide sort of further data collection that we might do with the 6800. Once we've identified those outlier lines, then we can apply the 6800 to really look at sort of detailed physiological response in those to try and evaluate sort of a mechanistic understanding of what's driving the response that we see with the 600. And I have an example data set here that was collected in a pepper experiment that we had the opportunity to participate in. And so in this experiment, the researcher had a large collection of wild collected pepper genotypes, we'll call accessions here. And for short, I'm going to call it 31 accessions. It's actually 30 accessions and 1 control commercial variety. But for the purposes of our discussion, that doesn't really matter. We'll just call it 31 accessions or 31 different lines. And we had the opportunity to participate in this experiment where they were looking at water stress or drought response in these peppers. And so there was a point at which they were ceasing irrigation and looking at how the physiology changes and how these things respond as the plants dry down on the end of irrigation. And so we were able to bring 2 6800s and a 600 to bear on these plants and collect some data. And it provides a nice example of this screen evaluate model. And so what my field assistant did was he started by collecting just diurnal curves of stomatal conductance under the current conditions, right? So just going around the greenhouse and measuring every plant in those at sort of a fixed interval. And that interval was about an hour to an hour and a half. It took him about 30 minutes to go through all of the plants. The setup was in replicate. So each one of these time points that you see on the plot here represents over 100 measurements with the LI-600. And what I have plotted from this in the gray symbols are the mean stomatal conductance for all accessions, plus or minus a standard deviation. And then the solid and the open symbols are 2 accessions that we pulled out from this for further physiological analysis. And I'll talk about how we did that. I'll start by saying, whether you're using this to screen some population to separate things, this is a really handy data set to have, because it tells you if you want to do measurements with the 6800 where you're looking at capacities, what period of the day you should think about doing those measurements in. In this case, we see that stomatal conductance peaks around midday and then it starts to tail off after that. And that tells me that if I want to go do some response measurements, I better get to the field and be done with them before midday, because if I go past that, then I'm fighting other things and I'm not getting a picture of what the real capacities are. For our selection of this, what we're calling insensitive and sensitive accessions here, and that's with reference to their drought response or water stress response. We picked that out based on the afternoon data. The one that we called insensitive and did for further analysis here was a clear outlier in terms of that afternoon response, thinking that the afternoon response in this case was going to be similar to the drought response because of, you know, high VPD, high temperature. It seemed like a good candidate. And our sensitive variety fell out on the lower side, but we had some sort of pre-existing evidence that it might respond very poorly to drought. And so it was one of the lower performers here, and it's the one that we selected for further analysis. So at this point in sort of the experimental scheme, we cut irrigation, and then we came back later to look at sort of how these things responded. And we did a reasonably good job of picking a sensitive and insensitive line. The insensitive variety did perform less than the mean in sort of mid-morning and late afternoon, but the midday conductance was well outside of 1 standard deviation from the mean, which was kind of nice. And our sensitive variety did turn out to be the worst performer in terms of conductance, at least across the entire collection of plants that we were looking at. And this on itself tells an interesting story. I mean, there's great value in stomatal conductance, particularly stomatal conductance across the course of a day. But because we have 6800s with us, we can dig deeper into this and we can look at what are the physiological ramifications or the physiological responses behind this change in stomatal conductance. And so the first thing we did was we took the 6800s and we applied them to do light response measurements, right? This is a good response measurement. It gives us information about energy use. And for the most part here, it was utterly useless, right? Other than to say that despite having lower conductance, there's no real difference in how these plants acquire carbon or how they use energy between the stressed and non-stressed condition, right? So we can think of NPQ and 5PS2 as giving us information about sort of energy use. It's maybe outside of the scope here to talk about why that is, but there are indicators and they're not really different between pre-water stress, post-water stress, and either the water stress sensitive or not sensitive lines. And light response carbon assimilation gives us information about carbon uptake capacities, and they're not really different either. But there's another way to look at this data that does tell an interesting story. And that's if we take that light response data and plot it, or the light response of carbon assimilation, and plot it against stomatal conductance instead of light. So this is the data from the same curves, just plotted differently. And what this plot shows you is something that's essentially intrinsic water use efficiency. And so it tells us a little bit about how the plant is regulating water loss in the context of carbon uptake. And we can see that in the pre-stress condition, both the sensitive and insensitive accession, they treat it basically the same. You know, the sensitive line, the data is a bit noisier, but you know, if you fit a straight line to that, it's not particularly different than it is for the insensitive variety. But as soon as we apply that water stress, we can see that the slopes of those lines steepen up. So they're being more careful about how they use their water for a given amount of carbon coming in. And the other thing you can see is that this sensitive accession shifts the curve to the right, right? It's much closer to a zero intercept. And so it's doing a better job at regulating water loss for carbon uptake. We did CO2 response as part of this using a new technique that's available in the 6800 called the dynamic assimilation technique. This allows us to do non-steady state curves. I'll talk about why we did it this way in a summary slide here in a second. But you can see the same deal. This is a post-water stress CO2 assimilation curve for both sets of our plants. And It's noisier on the sensitive variety because conductance is just so much lower, so that adds uncertainty into the calculation. But the thing to note here is that the carbon uptake capacity for that sensitive variety has dramatically increased. And so while stomatal limitation is greater because the stomata are closed down, the plant over a really short time period in this case has upregulated its ability to take in carbon to compensate for that, which is a pretty cool thing to see. And it's something that we wouldn't be able to see if we were just looking at conductance from the 600 alone, right? So we get not only a deeper picture, but we haven't had to put in as much effort to get a really neat story here. So this is my field assistant. You might recognize him from the opening. We call him Rod Porometry Madsen because over the course of this experiment, he did over 1,000 individual conductance measurements in 2 days. Pretty impressive. What we can say from this is that the stress-sensitive accession maintains carbon assimilation capacity despite the significantly reduced conductance. Like I was saying, if we're just looking at conductance and we ignore carbon here, it might lead us to the false conclusion, at least early on, that these plants aren't taking up as much carbon because the stomata are shut down. But instead, what the plant actually did was upregulate its capacity to take up carbon to compensate for the fact that the stomata were closing. The only way that we were able to see that was by doing response curves with the 6800. The downside is that these are inherently slow. So with 2 LI-6800s applied in this system, we were able to measure each accession in triplicate, right? So we could put some error bars on the data. It's a pretty important thing. And we were able to make triplicate CO2 response curves because we used the DAT technique on top of that. So each instrument, we were getting through 6 response curves a day. The light response curve was taking us maybe 45 minutes to get through. The CO2 response curves using the DAT technique were closer to 15 minutes or so. But if I wanted to do that on every accession that we were looking at, right, there's 31 of them here, or 30, depending on how you want to look at it. And with 2 instruments, I can get triplicate replication on 2 accessions. Well, if I want to do that on all of them, well, that's 15 days to do the 6800 measurements. Well, that's just impractical, right? I mean, what we were looking at here was a rapid drought response. What we saw 2 days after we cut irrigation and what we would see 15 days after we cut irrigation, that's totally not the same thing. They're both interesting maybe, but they're not the same. And with how strongly these plants had responded by the 3rd day that we were there, I don't think many of them would have been alive by the 15th day, right? So we did something that we couldn't have otherwise done on all of them, right? We did it on a subset that we selected using the 600. And we could do that selection because we could measure everything really quickly with the 600. Not only could we do it quickly and get a measurement on everything, but we could get multiple measurements over a diurnal time course to get an idea of the diurnal behavior. I would say our collaborators that we were there with were attempting survey measurements with the 6800 in prior to us arriving with the 600. And they were managing to get 1 measurement on each plant in a window that was about an hour longer on either side than what we were using for our diurnal time course with the 600. So that data was so confounded by time of day that you couldn't tease anything out of it because there's no replication to it. And you're measuring some plants at 8 o'clock in the morning and some plants at 4 o'clock in the afternoon. And it's clear to see from the 600 data that they don't behave the same. So this is my example of a screening evaluation kind of program. Let's look at a different use of the 600 with the 6800. And for that, we'll just use a simple instrument output model to discuss sort of an introduction to it, and then we'll talk through it. And the idea here is that for the instruments we use, we often look at the final outputs and assume that's something the instrument actually measured. Rarely, if ever, is it the case that that is solely the product of any direct measurement or is a direct measurement itself. Even the things that we often think about as direct measurements as sort of detailed users like us at LI-COR aren't really direct measurements, right? 6800 measures carbon dioxide. Well, but it doesn't really measure that. What it really measures is current. And we go through a bunch of hoops to get from current to a gas concentration. And so for anything that we see that comes out of an instrument, there's sort of this hierarchy of things that happen to it, right? So we might make some direct measurements. To those direct measurements, we might apply instrument constants. So this is like the idea of getting from a current to a carbon dioxide concentration. The instrument constants would be the instrument calibration. And that gives us some first level of outputs. And then we might pair those with some more direct measurements, maybe some more instrument constants, do another calculation, get another level of outputs, and so on and so forth, right? For things that are measured directly by the instrument or things that are instrument constants, these are well known by the instrument because either they're measuring it or we at LI-COR characterized it at the factory. But there are a number of places where we need user input to get the things that you want to know from the instrument. And for the most part, we make a first best guess at these, and then we rely on you as a user to parameterize them, tell the instrument the right value to get the right answer from it. And so I'm going to give an example where we use the 600 as a way of characterizing a user input. And I'm going to explain why we need that user input in the first place. So both the 600 and 6800 measure apparent transpiration to water vapor as their first output at getting to a second level output of stomatal conductance. So transpiration is our starting point for stomatal conductance to water vapor. And both instruments make the assumption that the transpiration they measure in the chamber is a diffusive flux, right? And so it can be described by Fick's first law of diffusion, which I show here, that says a diffusive flux X is a function of diffusivity along a diffusive pathway, right? So that's the distance between the dashed gray lines in my figure, and a gradient in concentration over that distance, or we could think about it as a diffusive conductance GT along the pathway times the difference in concentration between the two endpoints, right? So what we want to know to start with is GT, and we measure transpiration. So we plug some things in, we rearrange this. I've defined the endpoints here, what we call WI, the leaf intercellular water vapor concentration. We get this by leaf temperature and atmospheric pressure and the assumption of saturation. We measure WA as part of the mass balance that gets us transpiration in the first place, this value E. The term in parentheses at the top is essentially a dilution correction. Otherwise, this looks just like the equation I showed you on the other slide with meaningful values plugged in and rearranged for total conductance. Total conductance is great. However, it's total conductance along the diffusive pathway. And only one point in that pathway is under biological control. That's the stomata. That's the part we really want to know. So if we want to know what the stomata are doing, we have to partition the total conductance into the various components that are the biological one and the non-biological ones. And we do that by applying an Ohm's law model. And so I've just overlaid a simple resistive model following Ohm's law to describe the various resistance along the diffusive pathway, noting that a resistance is an inverse of a conductance and a conductance is an inverse of a resistance. So 1 over R equals G and 1 over G equals R. And we can define a resistance for the intercellular airspace, for the plant cuticle, for the stomatal aperture. That one's variable, that's why it gets an arrow across it, and one for the boundary layer around the leaf. And it's too much to know and solve all on its own. We have to make some simplifying assumptions. One, that intercellular resistance is small. That adds directly because it's in series. Add a small number to a bigger number, doesn't matter so much, we ignore it. The other one that we make the assumption of is that cuticular resistance is large to water. It is, it's not absolute, but it is large. And we add that as an inverse. So 1 over a large number is a really tiny number. So we pitch it. And we're left with this really simple model that says if we can measure the total conductance and we know something about the boundary layer, this is one of those instrument constants that we characterize at the factory, then we can get stomatal conductance. Life is good. Did I just undermine the whole reason that I'm talking to you? No, because the model that I'm showing here is one-dimensional. It applies to a single leaf surface. That's great in something like the 600, but in the 6800, we measure both leaf surfaces. And so to double this model and have it account for both leaf surfaces, we have to know something about how much conductance is coming from one leaf surface to the other. And the way we make that differentiation in the instrument is we replace that 1 over boundary layer conductance with a parameter we call Kf. And Kf is derived from K, the stomatal ratio, which is the ratio of conductance on one surface to the other. Okay, so how do we get K? Historically, this would be done by stomatal peels, where we count stomata on one surface of the leaf and count stomata on the other surface of the leaf, and then we make the assumption of some fixed correlation between conductance and density. We know that the fixed correlation assumption is not always good because there's patchiness in stomatal behavior, and we know that it is a tremendous pain to count stomata under a microscope to come up with this number. It takes a long time and effort, right? With the 600, we can measure each leaf surface independently. We can make a direct measurement of the conductances on those surfaces. And if we take the ratio of them, then we can get stomatal ratio. So it's pretty simple. We walk forward, we measure the leaf in its normal orientation. We flip the 600 over, we measure the top of the leaf with the instrument inverted, and that's all there is to it. So I'll show you a quick example about how much impact this actually has. And the example I'll use here is a field experiment where we were working in a hemp crop. And we just happened to have access to a field of hemp that were genetic clones, but those clones had been made and planted in the field at different times. So we had the opportunity to work across a physiological gradient of genetically identical individuals. And that gradient spanned from plants that were purely vegetative, had been recently planted, to plants that were just starting to initiate flowering, to plants that were in full-on floral development. And this particular hemp variety is grown for those flowers, right? And part of the experiment, this was a larger experiment, but part of it was we were there to collect ACI response. And so I'm just showing those as an example of some of the data that we got. They're pretty looking curves. They show some phosphate limitation at the high end, which is kind of a neat thing to see. As part of this work, my field assistant, the porometry Madsen, did stomatal ratio measurements. And I went back and recalculated all the data that I collected with the 6800 to look at how much that actually impacted stomatal conductance and some downstream derivations from that. And so the plot here is the stomatal conductance after correcting for the right stomatal ratio. So the default on the instrument is 0.5, or 2 times as much conductance on one surface versus the other. And the value on the y-axis is just the ratio of that correct stomatal ratio conductance and the default stomatal ratio conductance. And then what you can see is that there is a clear dependence there's a clear change in the value, as we would expect, as the stomatal ratio changes. And the further that ratio is from the default value on the instrument, in the case here, it changed with phenological stage ranging from 10 times as much conductance on one surface to the other to 20 times as much conductance on one surface versus the other. So up to a 10x difference from what the instrument default was. So the further that is from the default, the larger the error, and that the error scales with stomatal conductance. It's not a fixed bias. So the higher the conductance, the larger the error. Now, hemp has fairly large conductances to start with. It's pretty unusual to see values above 1, but it's not unusual in agronomic plants to see values from about 0.3 to maybe 0.8, 0.9. And in that range of typical agronomic plants, man, the errors here are 5 to 10%. That's reasonably significant and maybe significant enough that you'd care about it. I care about it here because that error in conductance propagates further downstream. So the same sort of logical series of assumptions that I showed for getting from an apparent transpiration measurement to stomatal conductance is the logical sequence that we make to get from carbon assimilation to CI. And so stomatal ratio affects CI doubly because it affects stomatal conductance to water vapor and stomatal conductance to CO2, which are both part of the CI calculation. And before you get too worked up here, this is the same kind of plot as I showed on the last. So the corrected CI versus the ratio of that to the default value CI. And I say before you get worked up because the errors here are excruciatingly tiny, right? We're talking about plus or minus 0.5%. Who cares about plus or minus 0.5%? Well, I do in this case because it's not a fixed error. It's an error that changes with CI. It changes in what I don't have a better way to describe than a playground slide shake, right? There's the ladder up and it swoops down. And the region where the error changes the most with CO2 concentration is the region of the curve that we fit for a parameter called VC max. And so despite the absolute errors being relatively tiny, they get magnified in the fitting. And so in this case, I'm showing a conglomeration of data for all the curves. And I went through and fit each one of those curves individually to calculate VC max. And the difference between the stomatal ratio corrected and stomatal ratio not corrected data ranged from about 0.5%, who cares, to as much as 8%. Well, I would probably care about 8% quite a bit. So probably not a case to justify buying a 600 on its own to just go measure stomatal ratio, but it shows a really useful case where we can take the 600, particularly if we're already using it in one of these screening evaluation applications, and we can get another layer of data out of it that improves the accuracy of the data we collect with the 6800. The point being that the data from any instrument is only as good as its inputs. And some of those inputs it knows, like direct measurements and instrument constants, and some of those things it relies on you as a user. And it can't do any better than waiting for you to put the right value into it. And I think that's what I have. It is. So we have time for questions now. If you want to read more about these things, we have a nice application note that Ian put together from a lot of this data, where he talks about sort of the underlying physiology that the instruments probe, the different kinds of measurements that you can get from the two instruments and how they relate to each other. And then in the existing version of the app note, he's included a case study that talks about this experiment, the screening evaluation experiment with the pepper accessions. At some point, we'll have this updated with a case study that goes through the one that I just went through looking at stomatal ratio. And at this point, I'll turn it back over to Rod.
Okay, thanks, Jason. Great talk. I think we're gonna try and stick around for a few minutes. Looks like we got some questions, backlogged questions in the Q&A. Let me just pick a couple one that stood out. Here. This was actually in the chat, Jason. Ron said, he was talking about the pepper experiment. Shouldn't we have looked at the daily curve and see which one drops? Not all plant lines will peak at the same time. And I don't know if that's enough information, but.
Yeah, and I would say we did. I condensed it down to try and make it fit nicely in one figure. We actually took that first day's worth of data and looked at all of them individually. And that's how we got to the two that we pulled out. They did, for the most part, all sort of start shutting down at the same part of the day. So and maybe that's to be expected because they were all grown so similarly in such similar conditions. At that point, it wasn't drought stress causing that shutdown, but sort of midday depression, probably due to higher VPD in this particular case. And we also had to cheat a little bit in this because we had a limited time window. So it wasn't entirely the 600 that we used to screen for these, although that was certainly a supporting factor in the particular accessions that we used, just because of the time limits of the experiment. All that data that I showed you, the triplicate response curves and the diurnal curves with the 600, that's 2 days of field collection between Rod and I. Hopefully that answers the question.
Okay, thanks, Jason. Just a quick comment for everybody here. You can elaborate a bit. Is the data analysis with the LI-600 difficult, or is it like the 6800 where an Excel file is formed? Can you comment on that?
Oh, yeah. It's a little bit different than the 6800. We don't make an Excel file with the 600 because there's no reason that you'd ever recompute anything. Right, it's a fixed leaf area, and if you're not measuring that leaf area, the instrument doesn't work. And it's a tiny leaf area. Let's see if Zoom will show it. This is this is the leaf clip. It's a little, it's like 7 millimeters in diameter. So you're probing a tiny spot on the leaf. But what we output is a text file that you can open in Excel and extract the things out of. We've done all the relevant computations, and just like the 6800 data files, we include every other ancillary parameter that you might care about or that we would care about should you ever need technical support on the instrument.
Okay, there's a lot of different questions here about.
I'm sorry, Rod, before you
Right.
go on, I'll throw in one other detail is that part of what allowed us to do such high density data collection with the 600 was that the 600 includes a barcode scanner built into it. And so we had every one of the plants in that experiment barcoded so that when Rod went through and made the conductance measurement, at the same time, he could hit a button and that would pull in all the metadata for that particular individual plant and pair it up in the data file, which really helps a lot with throughput, and it ensures that you have everything that you need in the final analysis.
Okay, this is a common question, and I know the answer is probably not going to be great, but how about succulent plants? Which instrument is ideal for that?
Yeah, for conductance only, you can get away with some thicker things in the 600. If you want to do fluorescence at the same time, there's some geometrical constraints that limit it sort of to a thinner broadleaf plant. So yeah, for succulent plants, the 6800 is probably going to be the answer most of the time, unless we're talking about something that's got a really thin succulent leaf.
How about begonia?
I think you'd be fine with begonia using both instruments. You might see some leaf damage from the 600 just because of the way that it compresses a hard surface on top of the leaf to push it down into the gasket. But if you weren't interested in making repeated measurements on the same spot, I don't think that would matter too much to you.
Okay, I've seen a couple questions that are sort of related to the difference between the 600 and 6800 when it comes to stomatal conductance. And I think even one person mentioned they're not the same. So it'd be good, I think, if maybe you could comment on, spend a couple of minutes commenting on why those two measurements are a bit different at times.
Yeah, this is actually an interesting point. There are a lot of differences, actually. If you give them truly the same thing to measure, then you get the same answer. I mean, we've done a lot of lab validation that shows that. But it's really difficult to really give them the same thing to measure. And so when we do that, we're using like laser cut perforated plates and whatnot. This is a really interesting data set here because that shows this really well. So hemp in this case is a great example of this, but other plants exhibit differing degrees of this that affect this as well, right? So let me back up a step, right? So I said the 600 measures the lower surface of the leaf. Typically, you could flip it over and measure the top, but it measures the bottom surface. The 6800 measures both surfaces, right? And this is why stomatal ratio matters so much for the 6800. You can see the adjustments here, right? When we put in the right stomatal ratio telling the 6800 how much conductance is coming from the bottom versus the top, relatively speaking. And 10, 12% changes in the values. At the upper end of this curve, these are the values that were measured near ambient conditions. And they were way different on the 600 when we made the measurement in the field. But when we put the right stomatal ratio in, man, they lined up beautifully. And they lined up beautifully here because particularly in these plants that are undergoing floral development, the later physiological stages, almost all of the conductance is coming from the lower leaf surface. And so they're measuring something that's very similar. Now, if they had been measuring something that behaved a lot like the default value in the LI-6800, this 0.5, well, they're not going to line up anyway, even if I put the right stomatal ratio in there, because now I'm measuring two surfaces in one instrument and one surface in the other instrument. The other thing that often leads to discrepancies between them has to do with just the environment around the leaf, right? So in the 6800, you control the environment. It's really difficult for us as people to perceive how a leaf perceives the world. And so often, we make small changes to the environment that our leaf might be sensitive to. When we measure it in the 600, well, we're not controlling the environment. It is what it is, and it's what the leaf was seeing. No response physiologically, we get the right answer. Now, we put a leaf in the 6800, it might have started out at that conductance, but because we changed the light level the leaf was seeing, or we messed up the VPD, or we screwed up leaf temperature because we made it hotter or colder, and then the stomata start to respond. Now, by the time the instrument stabilizes, it's not really the instrument we're waiting on, it was the physiology we're waiting on, and now we're at a different conductance than we were before we put the leaf in the chamber. And so you get different answers. There's also some issues around patchiness and sort of spatial averaging, right? Because at the 6800, you're measuring a larger diameter area than you are on the 600. And so it's really difficult to make sort of a direct comparison between those values because you have so much you have to constrain for. But general patterns of behavior should be the same. The utility of the data together, absolutely. But thinking that one is a substitute for the other is probably not a good way to approach it unless you're prepared and capable of dealing with all the things that could make them different. And we have a nice technical note that describes what those things are. And if you're interested in making a go of it, that would be a great thing to read first.
Okay, thanks, Jason. So we're going to probably answer a couple more questions and then end the webinar. And there are a lot of questions, and I will tell you that someone will get to your question and respond back to you probably in an email if we're unable to answer it. We obviously want to be respectful of everybody's time here and try to limit the webinar in general. But if you do have a question in the Q&A and we didn't answer it, either our panelists didn't answer it or Jason, we didn't get to it live here, somebody will address it. So we'll go ahead and try to answer a couple more questions. I did see one, but it just moved off my screen. Can you speak a little bit, Jason, about the IRT versus the contact thermocouple on the 6800? I think they're asking sort of why we don't use an IRT on the 6800. It's my response to that would be at the time of the 6800 development, the IRT technology wasn't as good as it is today, but it's something we're looking into for the future. Any other response around that, Jason?
Yeah, I mean, so yeah, to Rod's point, definitely at the time that we started developing the 6800, the IRTs just didn't perform well enough for this kind of measurement. Despite the fact that some people did use them, it was challenging. And so we stuck with a contact thermocouple as we always have. And it's a trade-off. There's sort of two schools of thought of it. A lot of people like the IRT because you're physically averaging across the field of view of the IRT. And if you match that well to the leaf area you're measuring, like we do in the 600, then you get a really nice average leaf temperature to calculate conductance from. The flip side is that average temperature that you get is a function of emissivity, which we don't actually know for a leaf. We make a guess that it's 0.98, and that's the value we use when we do the calibration. To that point, even calibrating the IRT, we have to calibrate it against some temperature controlled standard, and we need a standard with the same emissivity as a leaf. Well, we don't really know what the emissivity of a leaf is. We have a good guess. We have to make a standard with a known emissivity that we think is close to it. And so there are uncertainties that build up in that. I'm not knocking the IRT and the 600. It is really freaking awesome. But that argument has been used for a long time to justify why you would use a contact measurement with a thermocouple. And so that's what we do in the 6800. The contact measurement has its own deficiencies, right? Because we know that stomata can behave patchily across a leaf surface. And the larger the leaf surface that we're trying to measure, the more likely a single point measurement becomes non-representative of the total leaf surface, at least theoretically. Whether or not that actually happens in your plant material is hard to know without characterizing it. We actually do give you another way to treat leaf temperature in the 6800, and it's my preferred method for pretty much all measurements. And the data that I've shown here is all collected using what we call energy balance for leaf temperature. And so an energy balance is exactly what it sounds like. It's a balance of all the major energy fluxes from the leaf to derive a leaf temperature without contacting it or having to measure it. And we do that by looking at, by using the transpiration rate, the light intensity of the chamber, the spectrum of that light. So it works really well for our light sources, and some information on air temperature and turbulence. The full derivation is given in the manual if you want to read through it, but it provides a really nice value for an integrated average leaf temperature across the total leaf area in the chamber.
Okay, very good. Well, I'm going to ask one more question, Jason, and then we're going to probably try and end the webinar. Again, if you do have a question, could just type it in. We'll have a recording of that, and somebody will get back to you via the email that you provided when you registered. I think the last question here, and I think it's a bit general and it's probably highly subjective, but I'm actually going to combine two questions. Do you have any recommendation of measuring time or duration? I just lost the question because people are asking a lot of questions. But related to time or duration to complete a data set using the 600 and 6800. And then the other question is, how many measurements do you recommend taking per plant and how many times per day? And I know this is all dependent on the experiment the person is running, but just in general, maybe some comments around measuring time, duration, and number of measurements.
Yeah, it's a tough question to answer because there's no right or wrong answer. Well, I say that again. There's no right answer. There's probably certainly wrong answers, but it's hard to know what the wrong answers are until you know what the right answer is. And so I can't really provide any sort of direct answer to it. What I normally tell people is that you should conduct some sort of little study before you actually make your serious data collection. You know, I've shown a lot of data here, and not all of it is maybe all that useful. Maybe a light response curve on its own wouldn't have been useful in our pepper data. And if I'd known that going into it, I might not have collected them. True, the intrinsic water use plots from it were pretty interesting, but what I generally tell people, and this is another place where the 600 and the 6800 make nice buddies, is, you know, my approach to new plant material in an experiment is to go and do some of those diurnal curves, just like Rod did with the 600 and the peppers, and just get an idea for what the diurnal behavior looks like, because it's hard to know without measuring it what that's going to be, or to get an idea of sort of the range of values that you might see, particularly if you've never worked with that plant material before. And then once I do that, I like to collect some light response and CO2 response curves to just get an idea of how the plant behaves. That helps me figure out maybe what the most useful measurements are to make in the first place. Maybe I don't need to make a full light response curve if I'm interested in carbon. I could just do A max, or maybe the lower part of the light response curve is interesting, or maybe just VC max from the CO2 response curve is interesting. It's hard to know. And then once you have that information, then you can answer the question that Rod asked. So, but until you have that information, it's really hard to know. And my preference is to constrain the specific leaf that I make the measurement on and measure more plants as opposed to more leaves per plant. So in all of this data that I've shown here, we were really careful in constraining which leaf relative to the meristem that we were growing. So like in the hemp data, we picked the first fully expanded leaflet back, or fully expanded leaf back from the main growing point that was wide enough to fill the 2 square centimeter aperture on the 6800. And even though we could have measured smaller leaves that were younger on with the 600, you know, that was the same leaf that we collected all the 600 data on. So, yeah.
Okay, well, we're approaching an hour here, and I wanted to keep it within an hour. I did see one more question I think would be important for people to understand that have an interest in these products. So if you don't mind, Jason, one more quick question.
Yep, let's go for it.
The 600 and 6800, are these products affected by temperature? And the second part is, does the moisture content of the leaf introduce variance in readings?
Oh, yeah. So certainly, I mean, certainly they're affected by temperature. If you chuck them in a bonfire, they would not survive that. But For practical measurements in sort of human survivable temperatures above freezing, it doesn't really matter. I mean, they're designed to work in that we do all the necessary temperature compensations on the measurements internally to the instruments. And so the only temperature question then becomes a physiological one. How does temperature affect the physiology you're measuring, not so much the instrument? And then to the leaf water content, yeah, the conductance measurement fundamentally assumes that the intercellular airspace is at saturation. That seems to be a relatively good assumption to the point that the plants have lost significant turgor. And so for most plants that we would be measuring anyway for gas exchange, the assumption holds up well enough that it doesn't matter. It does matter if that leaf water content is on the surface of the leaf. I can tell you in those hemp measurements, Rod and I got a late start to the day every day because there was just so much dew there. We had to wait for the leaves to dry off before we could measure them in the first place. So there are considerations, and then there are things that are not considerations.
Okay, well, I wanted to end this about an hour mark, so I think we're gonna go ahead and end the webinar here in a minute. If your question is in the Q&A and it did not get answered, someone will get back to you via email. So we'll make sure that we get a conversation going on that question, and you're obviously introduced you to one of our support scientists in the process, probably. So I just want to give a shout out to Jason for his time, and all of our panelists, and also everybody that stayed on the whole time and attended this webinar. Again, appreciate everybody's time, appreciate the interest in the content, and we will get back to those of you that we were unable to answer the specific questions shortly. All right. Thanks, Jason. Thanks all the panelists.